Hi everyone, this is chapter 13 and we're going to be talking about chromosome structure and abnormalities in chromosome structure. So today in this brief lecture video we're going to talk about the anatomy of a chromosome. So we've discussed lots of different ways that we can look at genetic information. We have talked about sequences of DNA, we have talked about genes. Now we're going to talk specifically about chromosomes. We're also going to talk about some of the abnormalities we see in the structure of chromosomes. Then we're going to discuss how we look at chromosomes using something called karyotypes, and talk about cytogenetics, and then we'll finish off with a discussion of what happens when we have the wrong number of chromosomes. So first of all, if you remember from our previous discussions, chromosomes are DNA that has been coiled up or bound up so that it's ready to divide into developing cells via cell division. So remember, chromatin was sort of that loose, unbound material that we need to access the genes. So when we're going through the processes of transcription, we actually have to have access to our genetic information, which is in that loose, unbound form. But when we need to copy DNA and move it around the cell, we bind it up tightly into these chromosomes. So chromosomes differ in size, they differ in shape, and they differ in the number of genes that we have. So if you remember, humans have 46 chromosomes. That's our diploid number. Remember, you got 23 chromosomes from mom and 23 chromosomes from dad. So we have 23 pairs, which is a total of 46. 22 of those are, again, identical chromosomes, autosomes we call them. So we number those from 1 to 22. And then your last pair of chromosomes are your sex chromosomes. So again, depending on if you're male or female, depends on whether you have two of the same or two different chromosomes. So we're going to talk a little bit about the anatomy of a chromosome, and we're going to talk about a few important parts. So the centromere is the largest, what we call, constriction of the chromosome. So if you remember, in cell division, we had those spindle fibers that were reaching out and were attaching to the centromere, and the chromosomes were being pulled around and moved around the cell via their centromeres. So it also happens to divide the chromosomes into arms. Okay, so you can think of arms as just basically parts of the chromosome if you want to think of it that way. The other important thing to remember are telomeres, and we've also talked about telomeres too. Remember, telomeres were kind of like those protective caps that are at the end of chromosomes, and every time your cell divides, those telomeres get a little bit shorter and a little bit shorter. And once they are short enough, or they're too short, that's kind of a signal to your cell that you can't divide that cell anymore, that the chromosomes are too fragile. So here we go. This is a replicated chromosome. Okay, so keep in mind that we're looking at a replicated chromosome because of this, again, this distinct X form here, right? So these are two sister chromatids. So the centromere is here in the middle. And then what we see are the telomeres at the tips, so at both the top and down here again at the bottom. And then we are separating these into what we're calling arms, the P arms and the Q arms. So the P arm would be sort of above the centromere, and the Q arm would be below the centromere. And I'll talk about heterochromatin and euchromatin in just a moment. So the centromere position is one of those physical features of the chromosomes. So depending on where the centromere is, depends on what type of chromosome we have. So if the centromere is directly in the middle and we have two equal arms of equal length on both sides, we refer to it as a metacentric chromosome. If it's either slightly north or slightly south, so if we're creating one long arm and one short arm, we call it submetacentric. We call it acrocentric if it pinches off a small amount of material on one end. So if there's a little tiny bit of material on the top and then a very long arm on the bottom, we would call it acrocentric. And finally, it's telocentric if there's only one arm, if the centromere is at the very top. We don't have any telocentric chromosomes in humans, but it is found in some other species. So this is what it would look like. Again, this would be telocentric if you had absolutely no genetic material above the centromere. This is acrocentric, metacentric, and submetacentric. Okay, and again, it just refers to the length of the arms, basically, and where the centromeres are located, dividing those chromosomes into pieces. All right, so let's talk about how we visualize chromosomes. So there is a way that we can actually make a chart. It's called a karyotype. And karyotypes are incredibly important for medical analysis. So sometimes when patients present different syndromes or symptoms that doctors necessarily can't figure out what they're related to, they will send patients to a geneticist who will actually create a karyotype. And the karyotype can give us a snapshot of what our chromosomes look like. And we can look for abnormalities. We can see if there's anything wrong. So karyotypes will actually line up the chromosomes according to size and shape. And we use stains and dyes so that we can actually see and look at the different parts of the chromosomes. So places that take up a lot of stain, they are these dark bands of the chromosomes. They're composed of something called heterochromatin. Heterochromatin are highly repetitive sequences of DNA. It's the same sequence over and over and over again. These are non-coding regions. You don't find a lot of genes in heterochromatin. The light bands are where you find most of your protein encoding sequences, your genes. This is called euchromatin. So we would take our chromosomes, again, we would number them from the largest to the smallest, and then we pair each with its homologue, and this is what it looks like. And so we've got, again, chromosomes number 1, 2, 3, 4, and again, you can sort of see all the way down to, technically, chromosome 21 pair happens to be the smallest pair, but pair 22 is fairly small as well. And then this individual we know is a female because she happens to have 2 X chromosomes. And I just realized, I apologize, on the previous page here, it says 24 chromosomes. That should be 23 chromosomes, not 24. So please ignore that mistake. All right, so there's a whole subdiscipline within genetics called cytogenetics. And cytogenetics just focuses on chromosome variations. So cytogeneticists are looking at not only abnormal numbers of chromosomes or abnormal shapes of chromosomes and how these things can lead to different types of genetic abnormalities. So sometimes we have chromosome anomalies that cause a very distinct physical syndrome. For example, Down syndrome. That's having an extra chromosome number 21. There are a whole associated symptoms when it comes to Down syndrome, so physical syndromes associated with that. But there's sometimes that you will have slight abnormalities in chromosomes, and again, it doesn't present anything that is very obvious. It can be things that are hard or difficult to diagnose, for example, and that's where karyotypes can be used for lots of other things. They're not just used for medical diagnosis, they're also used to study families. So let's say we have a structural abnormality and we find it in an individual. We can actually get karyotypes from their parents, and if they're available, their grandparents, and we can find out if this structural abnormality is something that's inherited or if it was sort of a spontaneous mutation. We can use it for population studies. We can look at the effects of environmental toxins. Is this group of people who are exposed to this toxin, do their karyotypes, chromosomes show abnormalities, whereas the rest of the general population does not. And then evolutionary relationships. So you can compare banding patterns in different species. So a karyotype can be abnormal in 2 different ways: chromosome number and chromosome structure. As it turns out, abnormal chromosomes account for at least 50% of spontaneous abortions. So by that we mean miscarriages. So if a woman miscarriages, half the time that's due to something wrong with chromosomes. Too many chromosomes, too few chromosomes. There are a whole host of reasons that can occur. Because we have better technology at this point in our medical history than we have ever before, more and more people are being diagnosed with chromosome abnormalities, whereas before we might not have known what was wrong with them, and they sort of just would have gone through life with some symptoms or syndromes that they couldn't explain. And now we actually can by looking at these karyotypes. So let's talk about structural abnormalities first. The first one we're going to talk about is pretty simple. It's a deletion. So if there's a break in the chromosome and a piece goes missing, it's called a deletion. A duplication would just be the opposite. If there's a section of the chromosome that gets copied more than once, we have duplicated genetic material. So if we're looking at a karyotype, then this chromosome would be too short for a deletion, and this chromosome would be too long for a duplication. Depending on the size of the deletion or the duplication, depends on if there will be what we're calling an associated phenotype. So if you miss just a very small amount of genetic information, especially something like heterochromatin, which just is a repetitive sequence, it's probably likely that you're not going to see any symptoms. Likewise, if you make a duplication of that information, it probably isn't going to have an impact. But if you have a large sequence of genetic information that you're duplicating, then you might have to start to see some types of, again, a phenotype. And when we say phenotype, we just mean physical expressions of those duplications. So you might see things like curved spines or learning disabilities or autistic features or seizures. There's a whole host of things that could be explained because of the duplicated sequence we see. Other things that can happen incur inversions and translocations. So a translocation is where two chromosomes that are not homologous, these are non-homologous chromosomes, they exchange parts, okay? And not, remember in meiosis, we had homologous chromosomes which were exchanging information and changing pieces, and that was okay. You don't want non-homologous chromosomes to do that because then you're putting genes where they don't belong. And an inversion is very simply where we have a break in the chromosome, but instead of the piece going away, it gets flips upside down. Okay, let's do a quick comprehension check. Which of the following terms describes the exchange of chromosome segments between non-homologous chromosomes? Is it a non-disjunction, a translocation, a disomy, an isochromosome, or an inversion? It's a translocation, right? So when we are swapping out information on two non-homologous chromosomes, we refer to that as a translocation. All right, let's talk about other ways we can see abnormalities in karyotypes. There's something called polyploidy, and in polyploidy, you would have an entire extra set of chromosomes. In humans, this leads to a spontaneous abortion, again, a miscarriage or a stillbirth. No individual can survive this condition. You cannot have an extra set of chromosomes and be a normal functioning human being. Your body just can't do it. However, polyploidy is common in other species, especially plants. Plants do this a lot. They will actually have 3 sets or 4 sets or multiple sets of chromosomes, and it's something that plants are able to, they can balance it out so that all of those extra chromosomes are not impairing their functions in any way. So there are certain species where polyploidy is okay, but again, for the majority, the vast majority of living things, especially animals, polyploidy can't happen. Aneuploidy is something that's a little bit more common, and aneuploidy is when you have chromosomes that are either missing or where you have an extra chromosome. And depending on whether you're missing or have an extra one, depends on whether we use which terminology here. A monosomy is when you're missing 1 chromosome, and a trisomy is when you have an extra chromosome. So this is caused by something called non-disjunction during meiosis. So again, the vast majority of time, trisomies and monosomies produce inviable embryos. It means you can't survive that condition. However, there are very few instances where you have individuals that can be born who are missing a chromosome or who have an extra chromosome. So non-disjunction is simply the failure of chromosomes to separate normally during meiosis. So remember, during meiosis, we had those homologous pairs of chromosomes and they separate. Okay, well, what happens if they don't separate? What happens if they stay together? Well, this is where you get an instance where you have either an extra or a missing chromosome. So depending on when the non-disjunction occurs, depends on how many extra copies we'll have. So if it occurs during meiosis 1, we have copies of both homologs in one gamete. And if it occurs during meiosis 2, you have both sister chromatids in one gamete. So let me show you kind of what this works. So let's say this happens in meiosis 1. So here's our homologous pairs of chromosomes. They did not separate the way they were supposed to. So what happens is, in this case, it's a little, well, it's not confusing. We're looking at the male, the sperm here. So what happens is, as the sperm is being produced, we have spermatocytes here that have, again, an entirely extra chromosome. So when those sperm are developed, then they have, again, 2 copies of this particular chromosome, whereas these sperm have no copies of that chromosome. So if we had a fertilization event here, we would have, again, individuals that are either trisomic, which means they have an extra copy, or monosomic, which means they have, they're missing a copy. If it happens during meiosis 2, it's a little bit different. So we have meiosis 1, we split the homologs up. Meiosis 2, we did not split the sister chromatids up. So again, now you will have a greater chance of 2 what we will call normal zygotes, and then you would only have 2 zygotes then that would either have a monosomy or a trisomy. So here's kind of a nice summary of all of the different types of chromosome abnormalities we just discussed, including polyploidies and aneuploidies, and the deletions, duplications, translocations, and inversions. So this is a really nice summary of all of these conditions that we just talked about. And then I do just kind of want to briefly mention a little bit of what's called chromosome shorthand. So when we are discussing individuals and we're talking about them via their chromosomes, we use a little bit of an abbreviation to sort of discuss an individual. So an individual who is completely normal, who has the correct number of chromosomes, we would have the number 46. That indicates the number of chromosomes they have. And then after that, we put their sex chromosomes. So a normal male would be 46XY, a normal female would be 46XX. If they have a syndrome of some type, an aneuploidy, a polyploidy, a monosomy or a trisomy, we can indicate that with their chromosomes. So an individual who's missing an X chromosome, she has something called Turner syndrome, and she's 45X. A male who is a trisomy, who has an extra X chromosome, is 47XXY. And you can kind of see as we go down, these are, again, having extra chromosomes. We can even abbreviate individuals who have missing parts. So this is a male who happens to have a deletion on the long arm of chromosome number 7. Okay, so again, not to get too caught up in this chromosome shorthand, but it's sort of an easy way to be able to talk about, again, these chromosome abnormalities and sort of abbreviate them. So the one aneuploid condition I am going to spend a little bit of time talking about, because it happens to be a more common condition, is Down syndrome. So 47, and then you would use plus 21, because that's the extra chromosome that we have. So this is just X, and then it can be XX or XY depending on the individual. So it affects about 1 in 1,000 newborns. And there are actually 2 different ways you can get you can have Down syndrome. You can actually be a trisomy individual, where you actually have an extra copy of chromosome 21, or we also see Down syndrome happening when there's a translocation between chromosome number 14 and 21. There's always some degree of mental retardation. The other things that these individuals face are typically heart defects and differing facial features. So people with Down syndrome have a shortened lifespan. They typically live between 50 and 60 years, and that's mostly, again, due to the heart defects that they have. And what's interesting is that for both males and females, now it's not just women, but we know that the older the male or female is, the greater the chance there is of having a child who has Down syndrome. And it's just because that process of non-disjunction in meiosis is more likely to occur the older that you get. Okay, let's do another quick comprehension check. A male has trisomy 21. What is the correct shorthand to designate this condition? So I'm not going to read all these, but you can look at the answers 1, 2, 3, and 4. So which one is this individual? It's actually number 3. So he has an extra chromosome, so he would be 47. He's a male, and the extra chromosome that he has is chromosome number 21. Okay, there are the important terms and concepts to know after this chapter.